Hello, my name is Sean McIlwain. I work as a scientist at the University of Wisconsin-Madison School of Medicine and Public Health. My talk is entitled MASH Native, a Universal Tool for Native Mass Spectrometry and Top-Down Proteomics. Top-down mass spectrometry is a powerful method for comprehensively characterizing proteoforms, which arise from genetic variations and post-translational modification. However, a paucity of data analysis software designed for top-down MS is recognized as one of the major barriers to utilizing top-down proteomics. Native top-down MS is even more underserved than denaturing top-down due to challenges in deconvolution of low-resolution MS data and poor fragmentation efficiency than denaturing top-down MS. While a number of software tools have been developed to address certain aspects of native top-down MS data analysis, individual tools are limited in scope and MS file type compatibility. To address this challenge, we present MASH Native as a universal and comprehensive software environment for native top-down proteomics. The MASH family of software tools has been developed over the course of 8 years. Initially, MASH Suite was released in 2014, allowing the analysis of a single protein using MZ-XML data. In 2016, MASH Suite Pro was released, which included additional support for thermal data and searching for multiple proteins. Around 2017 to 2018, we received NIH support to further improve the software by extending the number of raw data formats supported and data analysis algorithms provided. In 2019, MASH Explorer was released, which provided a universal software environment that supports many more algorithms. In early 2022, MASH Native was released, providing further functionalities to process native mass spectrometry data. We continue to improve upon MASH Native to make it more comprehensive and usable for the top-down proteomics community. Written to run within the Microsoft Windows environment, MASH Native is a descendant of MASH Explorer and includes multiple vendor format support, integration with many algorithms for deconvolution and database searching, and several additional features. Currently, MASH Native supports Thermo, Brooker, Waters, MZXML, MZML, and MGF files as input into the system. MASH Native can import experiment files by using the vendor libraries directly or through the use of ProteoWizard. The software supports a number of deconvolution algorithms, including eThrash, TopFD, MSDconv, pParseTD, ProMex, FlashDconv, and UniDec. For database search, we support MS-Align Plus, Topic, pTop, and MS Pathfinder T. In the background, MASH Native performs the necessary conversions between upstream and downstream tools and automatically imports the results back into MASH Native, allowing the user to visualize and interact with their results. MASH Native provides two main modes. Discovery mode allows users to analyze LC-MS data to identify, characterize, and validate proteoforms. After data import, the user has access to the chromatogram and mass spectrum views. Process Wizard allows the users to choose which deconvolution and search methods to use when analyzing their data. Through use of the workflow manager, multiple analyses can be queued to run and may be run in parallel depending upon the number of cores available. Search results can be investigated and each proteoform can be characterized in the sequence panel. The other mode, targeted mode, provides tools to further characterize a single proteoform. After importing the MS scans for a single proteoform and the protein sequence, the user can investigate the observed deconvoluted ions which have been mapped to the theoretical masses from the proteoform of interest. MASH Native provides additional tools, including an ion finder that can help with the discovery of putative ions from a proposed proteoform that may have been missed by the deconvolution algorithms. This slide shows a layout of the main interface for MASH Native. The workflow and parameters panel controls the data import and processing steps. The results panel can display the mass spectrum and chromatogram and has tools for exploring individual scans within the data set. The sequence table provides the ion fragment map on proteoforms that have been matched to the mass spectrum through the database search. The mass view contains the information on the peaks, the deconvoluted masses, and the database search results. The experiment list view shows the current mass spectrometry data feds that have been loaded into the system. We'll now talk about other features that are available to better assist the native MS community in analysis of their data. The red boxes show some of these features are accessed. UniDec has been incorporated in the suite of deconvolution methods, which significantly improves our native top-down handling abilities. Match Native provides both automatic and interactive modes to allow users flexibility in running UniDec and within Match Native. By editing the deconvolution parameters or selecting preset parameters in interactive mode, the resolving power of the deconvolution can be matched to the resolution of the MS data to provide low resolution deconvolution or high resolution deconvolution with isotopically resolved peaks. In addition to importing the found deconvoluted peaks in the master charge space, Match Native also imports UniDec's charged deconvoluted spectrum for visualization. This function is available in Match Native's targeted mode. Future versions of Match Native will provide further functionality within this display component. Match Native also allows users to perform spectral summing in a variety of different ways, including several options for selecting which scans to average, which algorithm to use for performing the summing, and how to handle MS1 scans. This feature is particularly useful in native MS applications when trying to improve signal to noise by combining multiple scans either as a grand average or for MS precursor-based summing. Match Native provides the ability to search both terminal and internal fragments. To help with visualizing the internal fragment matches on the sequence table, we have also provided heat map coloring of the amino acids to indicate the amount of fragment coverage. Match Native also allows the overlaying of the matched terminal fragment markers on top of the coverage heat map. Matching internal fragment ions can greatly improve sequence coverage for native MS applications, enabling sequence interrogation in regions that are typically difficult to fragment, such as disulfide bonded regions or portions of the sequence protected by tertiary or quaternary protein structure. We're continuing to develop Match Native to further improve the breadth and depth of its functionalities. Here are a few of the features that we plan for the next major release of Match Native, which is slated for late fall of this year. In particular, we are focusing on providing quantitation methods using label-free MS1 data. We are planning on providing this functionality internally, as well as looking and importing the quantitation results that are available within the set of supported tools. I'd like to thank our collaborators, the past and present members of the Match team, the Dr. Inga lab members, and our funding sources. Match Native is available through our website and has been downloaded by users all over the world. And we have many avenues for users to communicate with the Match team, including a forum, Twitter, and email lists. I'd also like to mention that Match Native is actively maintained and continually improved upon to be an indispensable tool, and we appreciate your feedback. Thank you for your time, and I hope you consider giving Match Native a try.